So, good morning, students. We are going to study FSH test, and in this test, here the aim is to determine FSH level in blood. What is FSH stand for? So, we know here, and the method here we are going to know is ELISA, that is sandwich ELISA. So, this is the full form of ELISA: enzyme, E for enzyme, L for linked, I for immuno, S for sorbent, and A for assay. Enzyme, linked, immuno, sorbent, assay. So ELISA that we are going to use in this technique, in this test, is a sandwich ELISA. It's a special type of ELISA where the FSH is sandwiched between two specific antibody. Here the full form of FSH we are going to know is a follicular stimulating hormone. F for follicular, S for stimulating, and H for hormone. So this is the aim to determine FSH level in blood, and the method that we knew is ELISA method. Let's know some general information about FSH. This FSH is secreted by anterior pituitary gland and basically produced in gonadotropic cells of anterior pituitary gland. The production of FSH is regulated by gonadotropic releasing hormone, which is released by hypothalamus. So hypothalamus, release this gonadotropic releasing hormone, and this hormone acts on gonadotropic cells of anterior pituitary gland, where FSH is produced. The FSH, once they are produced, they act differently in male and female. In male, FSH acts on the Sertoli cells present in the seminiferous tubules of the testis. So the testis specific cell is the Sertoli cells, which is the target cells for FSH, and this specific cells causes spermatogenesis. The production of sperm takes place in the Sertoli cells, and this FSH also stimulates the synthesis of inhibin hormone, which further inhibit FSH secretion. So inhibin is the hormone produced in the testis, and this inhibin further stop the secretion of FSH from anterior pituitary gland. In women, this FSH act on granulosa cells of ovary. So ovary has a granulosa cells where the FSH stimulate growth and maturation of ovarian follicles of ovaries. So ovaries has ovarian follicles, right? And this ovarian follicles is grown and matured with the help of FSH, and further it produces eggs and progesterone. So eggs is a gamete, female gamete, and progesterone is one of the steroid hormones. This both are produced in response to the follicular stimulating hormone. So near ovulation time, at the time when the ova releases, this is about the mid of the menstrual cycle. So menstruation cycle, like 14th day, nearby 14th day, where the ovulation time takes place, and at that time the FSH level become peak at highest level, and we are going to know some chemical characteristics of FSH, where FSH is a glycoprotein hormone, which means it's a complex protein made up of protein and carbohydrate moieties. So we call it as a glycoprotein hormone. FSH has two subunits, namely alpha and beta. So there are two subunits present in the FSH. They are named as alpha and beta subunits. The alpha subunit is similar to TSS. This is LH, right? This is LH. So the alpha subunits is similar to TSS, LS, and human chorionic gonadotropin hormone. The three hormones, the alpha subunits are, similar to FSH. And the beta subunit here is the beta subunit of FSH is unique to FSH. right? The sequence of amino acid in beta chain is very, very specific to FSH here. Now, clinical significance, we are going to know that FSH measurement helps in the diagnosis of infertility in male and female. Some male and females are infertile, so that can be diagnosed with the help of FSH. This test is done along with uh, uh, this FSH test is done along with LH, right? So LH that we studied in the previous experiment, LH and FSH both are done together. FSH levels are elevated after menopause, when there is a menopause in the female. So FSH levels are remarkably increased. Castration is a procedure where testes of the males can be removed. So when there is a removal of testes, castration process. So after the castration, FSH level may be increased. And in premature ovarian failure, so when their ovaries are premature and there is a failure of ovary, then FSH level is increased. Increased serum FSH levels can be can be seen when there is a hypogonadism, means the gonads are they are not there is a failure, we can say the failure of the gonads. In such condition, FSH level increases. Primary ovarian failure, we say, or polycystic ovary disease. So this is a very, very specific disease which takes place in the ovary. And in such polycystic ovary disease, the FSH level comes at higher level. Primary testicular failure. So here, the failure of the testes, unable to produce sperm and testosterone. So here, the FSH level may be higher. And this is a chromosomal disorder, Klinefelter syndrome, where there is a double XY sex chromosomes present in an individual, and in such condition, FSH levels are very, very high. Now, decreased serum FSH level can be seen when there is a dysfunction of pituitary or hypothalamus. So, pituitary glands and hypothalamus are not functioning properly at that time. FSH levels are supposed to decrease. There are some symptoms which are noted into the by the the person who are infertile, or where the FSH levels are abnormal. Failure to conceive. So when the woman is unable to conceive, unable to get pregnancy, so maybe at the time we need to go through the FSH test. Irregular menstrual. So women with irregular menstrual should get the FSH tested. No periods at all, so there is no menstruation cycle at all, so those female can also get this FSH tested. Low testosterone level in the man, the person who has low testosterone should also get their FSH tested. Low sex drive in a man, so unable to do or have a feeling of sex drive in a male, so FSH level may be abnormal. Low muscle mass in a man, so the muscle mass, the muscle ratio with the body, is lower. So it's such patient, such male, the FSH levels may be abnormal. Now let's know about working principle. Here the given is wells coated with anti-FSH. So antibody coated wells are there, which is available. Now samples containing FSH where we need to perform FSH test. And the third antibody is HRP enzyme. HRP is a horseradish peroxidase. So this is the enzyme level antibodies, anti-FSH antibodies. So we have two antibodies. One is coated with the wells, and another is labeled with the enzyme. So and this is the sample where we need to determine the level of FSH. And this the method we studied today, the sandwich ELISA, where this FSH sample FSH is sandwiched between the coated antibodies and the conjugate antibodies. Okay, so here the first of all FSH present in the sample forms the sandwich between two specific antibodies, right? And then after that, when unbound FSH and HRP conjugates are washed off, now, upon the addition of the substrate, we get a color, and the intensity of color is directly proportional to the concentration of FSH in sample. So here, more color intensity, it means more FSH in the given sample. A standard curve is prepared. So we can prepare a standard curve relating to color intensity and the concentration of FSH. And from that standard curve, we can be able to find out the, the level of FSH in the given sample. Okay. So standard curve is a curve, okay, which is prepared by known concentration of FHS. So different known concentration of FHS can be used in the protocol, and we can get their specific absorption, okay, because of the difference in the color intensity. So absorbance versus concentration graph can be plotted, and that graph will be called as a standard curve graph. Okay, and by measuring the FSH in the sample, we get the absorbance, but we need to know the concentration. So by knowing the absorbance, we can just using the standard curve graph, the absorbance we can use, and we can find the concentration of that given sample FSH. Here requirements are microwells we require should be coated with the antibodies, basically alpha subunits. Antibodies are coated with to the microwells. Then we need a FSH standard. We require 6 vials with the concentration of 0, 5, 10, 25, 50, and 100 micro international unit per ml. So this is the the different concentration of the standard, okay, which can be used to prepare the standard curve graph. Now FSH conjugate reagent, where we can we should have an antibody coated with a conjugate, that is an enzyme. So here anti beta FSH, so beta subunits antibodies, which is coated with the HRP, can be used as a reagent. And we require a substrate. We call it as a TMB substrate, also known as tetramethylbenzidine, TMB substrate, where we have H2O2 and TMB. The concentration is 0.26 gram per liter of the TMB. Stop solution we can prepare by making 0.15 molar per liter sulfuric acid, and we can have a wash buffer. This is the wash buffer, which is concentrated type. Here we take phosphate buffer 0.2 molar as a wash buffer here. So HRP it means first it is peroxidase. Now materials required but not provided with the kit: distilled water, precision pipette, what we call auto pipettes, micro pipettes. Disposable pipette tips can also be required. ELISA reader, where we can absorb the intensity of different wells at 450 nanometer, 440 nanometer. Absorbent paper or paper towel can be taken to absorb the wells or plot the wells. And graph paper is required, where we can prepare the standard curve graph. And we require timer, so that the time is very, very important to this particular test. And we should give the specific time, proper timing for incubation. There should not be any delay in any steps while doing the procedure. Are you able to understand? Just give me feedback.
Yes, sir.
Okay. Let me check. Yeah. So this is the required materials which are not provided, right? And this is the microplate reader. This one is a microplate with the wells, right? The holder with the micro wells. And this is micropipette, this is tips, and this one is a tip with a box, right? So here, this is the holder with the micro wells strip, right? This is a way we can arrange our micro wells in the holders, and we can label each wells as a standard, controlled, sample, okay, or blank. Specimen collection and preparation. Here, what specimen is required? So the serum can be used in this assay. Do not use hemolytic or icteric or lipemic specimen. And please note, samples containing sodium azide should not be used in the assay. Why do we avoid sodium azide? Because this inhibits HRP enzyme activities. HRP enzyme that we use in the conjugation of antibodies. So that HRP enzyme activity can be inhibited because of sodium azide. So it should not be used in the sample. Specimens can be stored for up to 5 days at 2 degrees Celsius to 8 degrees Celsius, or up to 1 month at minus 20 degrees Celsius. So this is the specimen collection, preparation, and storage. Now we can dilute the buffer if it is 20x. So in such way we can make it 25 ml by mixing of the 20x 25 ml this wash buffer into 475 ml of this water. So here the wash buffer will be 20x. So this is the ratio we can make it. Stored at room temperature. Once we prepare the 1x wash buffer solution, then we can store them at the room temperature. Now procedure. Now before going to the step-by-step procedure, this is the general remarks that you should keep in the mind. That bring all the reagent specimen to the room temperature before use, and all reagent must be mixed without foaming. So prior to starting the procedure, that you should bring all the specimen at room temperature. You should mix them properly. There should not be the foam formation. All steps should be completely done without interpretation. Right? So there should not be any delay. And always use new disposable tips for each standard, control, and sample. So whatever you are taking, either different standard concentration or control or sample. So you always use new tips, and the tips should be disposable tips. So this is a way that we can take a multi-channel pipette that we can dispense, the reagent at a time in all the wells. So, or we can use a single channel pipette where one by one wells can be, added with the sample or reagents. The procedure we can start from here, that like place desired number of microwells in the folder. Desired number means how many microwells do we require. So we should keep them in the folder, and we should label them. The first well it belongs to first standard, second wells become to what that is what should be known and planned accordingly. So it should be labeled. Each and every well should be labeled, okay, and it should be placed in the folder. Now second step is 850 microliter of FSH standard, control, and specimen into the designated wells. So whatever the labeling we has done, so in standard all the standards, then control, and then specimen wells should be added with the 50 microliter of this volume. Okay, so this is the volume required to be added into the designated wells. Then after that we need to add 100 microliter of conjugate reagents. That is what we say antibody enzyme coated antibodies, right? Then we need to shake for 10 to 30 seconds. This is very important because this helps to mix properly, and should be done gently. Shaking should be done very gently. Now cover the plate, and now we should incubate them for 1 hour at room temperature. 1 hour incubation is required, and remove liquid from all wells after 1 hour. So whatever the liquid we have kept in the wells, we should discard them, right? And then we should go for the washing procedure. The 3 times washing is done using 1x wash buffer, and after that, at the last, we should do the blotting. We should absorb the wells with the blotting paper or absorbing paper. Then 800 microliter of TMP substrate to the wells. So TMP substrate should be added to the each and every wells, and then we should incubate now in dark. So dark, this is the dark incubation because TMB is a light sensitive here, but TMB we are adding 100 microliter. So after addition of TMB, incubation is required and should be done for 15 minutes at room temperature, but should be provided dark condition. And here after that we should add 100 microliter stop solution, then shake the plate gently so that it can mix, and ensure that that the color, the blue color changes to completely yellow color. So when we add the stop solution, the color which is blue color changes to yellow color here. And at the last we need to take the reading at 450 nanometer, and this would be taken within the 5 to 30 minutes of what we have done the added the stop solution. So after adding the stop solution within the 5 to 30 minutes, you should be able to take the absorbance of each wells at 450 nanometer. Right. So here some error analysis that should be done. The pipetting of all standards, sample and control should be completed within 3 minutes. When we add standards, when we add samples, when we add control, so ensure that you have pipetted them within the 3 minutes. And it is recommended that the wells to be read within 15 minutes following addition of stop solution. So here the 15 minutes is the maximum, but we can extend it then to 5 to 30 minutes. Okay. There should not be delay after addition of the stop solution. What it means. Standard curve graph, how can we prepare them? So this is the first, the standard graph is constructed as follows. Check FSH standard value on each standard vial. So what is the standard concentration on each vial? So it starts from 0. Okay. So each vial has a specific standard concentration. To consider the standard curve, plot the absorbance value in vertical y-axis. So absorbance, what we get after from previous slide. So the absorbance we should plot in the y-axis, and the concentration we should plot in the x-axis. So draw the best curve through the points, and do the absorbance for control and each known sample from the curve. So now, if you have plotted the standard curve graph, now you can be able to know the value of control and unknown sample if you have provided the absorbance of control and absorbance of unknown sample. Record the value for each control and unknown sample. So now we can be able to record now. If you get the value of the this control and unknown sample, we can be able to record. You can give the value of sample as a report to the patient now. Any diluted sample must be further corrected by the appropriate dilution factor. This is what is required, that if the patient concentration of FSH is very, very high, so we need to dilute the sample and then we need to proceed the procedure. And after, when we get the results, so the results should be multiplied with the dilution factor. Right? This is how we can get the different color in the different tubes, different wells. Right? Here, this is the standard curve graph, how it shows that this is the first standard concentration 0, and OD of at 450 nanometer is, we have taken 2 times OD, so 0.05, 0.0010, and average of this OD is 0.007. So what we consider that we have taken the first OD and second OD of different concentration of FSH, and this is what we have got the average OD of each concentration. So this is what we have considered here. So 0 concentration vial has OD 0.07. 5 micro international unit per ml has the average value of average OD is 0.095. 25 micro international unit per ml, the OD is 0.272x7. This is how, this is the concentration and this is their average absorbance. Now we can plot the graph. We said that on x-axis we can put the concentration, and on the y-axis we can put the absorbance. So you can see here that 0 concentration, the OD is 0.07. So first OD is 0.07. At the 5, the OD is 0.095. Okay, and this is 0.2 at 7. This is showing the concentration of 25, 25, approximately 25. At 50, the concentration is 0.670. At 100, the concentration is 1.477, and at 200, the concentration is around 2.584. So this is the concentration. So at concentration versus the OD, and then we can have a plot. This is a linear curve graph we have got, and from here we can be able to find out the concentration of unknown. Let's say the concentration of unknown is 1. So here we can put the point, and with the help of a scale we can line a draw. We make a line over here. So from here to here, this is the line we have done cutting, and from here we can make another line and we can find the value of unknown sample. So approximately 50 to 100, between this the value becomes around 75. So unknown sample value becomes 75 micro international per ml. So this is how we can do. Normal range in male, it is 1 to 14 micro international unit per ml, right? And then in female, the values of this FSH varies. Like in follicular phase, it is 3 to 12 micro international ml per ml. Then mid cycle, like mid cycle of ovulation cycle, it's a ovulation time where the value is 8 to 22, and at the luteal phase the value is 2 to 12. So in the female, at a different phase of menstrual cycle, the value is at the different levels, and this is the menopause where the value becomes very very high, 35 to 115 micro international per ml. So this is what we have learned about the experiment follicular stimulating hormone, how to do the test, what is the principle, what is the significance, what are the normal range, right? And we have also studied some chemical property of the FSH. We have come to the end of the slide. Now let's have a discussion. If you have any question, any doubt, we can do the discussion over here now. Let's discuss now.